episode 296 of the Stem Cell Podcast, Cardiovascular Development, with Dr. Maneesha Inamdar. Hello, everybody. We are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. This podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. For our guest today, we have Dr. Maneesha Inamdar. She's on the podcast to talk about her work using comparative models of developmental biology. We've also got our usual roundup of recent highlights in stem cell news. That's coming right up.
But first, when this airs, we'll already be in Hong Kong for ISSCR 2025. We can't wait to attend the meeting and see the latest advances in stem cell research. Visit isscr2025.org to plan your conference and see which sessions to attend. If you missed it, our previous episode featured a discussion on the ISSCR's new best practices for the development of pluripotent stem cell-derived cellular therapies, so be sure to check that one out if you haven't already. We'll get right into it here on the roundup for this ISSCR tangent episode. I mean, we're, of course, super excited about ISSCR. Just down the road, just a couple days, from now. One of the folks who may or may not be there, I actually don't even know if he's going to be there this year, is my PhD mentor, Dr. Joseph Wu, extraordinaire aficionado of all things iPS-derived cardiomyocytes and cardiovascular cells. This is a really beautiful study published in Science very recently from somebody who I know very well from my time in Joe's lab. First author here is Oscar Abules, and second author here is Huaxiao Adam Yang, who actually now also has his own lab at the University of North Texas in Dallas, I believe. This is, when you think of Joe Wu, a lot of what you think about is drug toxicity, drug screening, and using iPS-derived cardiac cells for that application, the whole clinical trial in a dish approach. But Joe is not traditionally known as a cardiac developmental biologist. I mean, right next door to him is Sean Wu, who's actually my other PhD advisor. I was co-mentored, and Sean is, a well-established cardiac developmental biologist, had one of the, early cardiac development cell papers using NKX 2.5 reporters to look at a bipotential cardiac progenitor. So Joe and Sean are a great team, but here Joe is foraying into the area of cardiac development and making a really awesome model system comprised of vascularized human cardiac organoids. And this is also sort of like a bipotential system that has the ability to turn into liver as well as cardiac. So the title here is Gastruloids Enable Modeling of the Earliest Stages of Human Cardiac and Hepatic Vascularization. This is really important across organoid biology, right? We want to create organoids that are, of course, self-organizing 3D structures, mimicking key structural functional characteristics of the in vivo organ counterparts. That's the definition of an organoid. But we want to create organoids that are better, and in particular, that are vascularized. And there have been a lot of approaches to this, engineering vasculature in there. The dream is to kind of organically create the vasculature during the differentiation process. And part of this is to, one, avoid, of course, necrosis in the core of the organoids. That's sort of preventing these things from getting big, because the bigger they get, the more likely you have necrosis in the middle of the organoid. So the vasculature theoretically would be able to deliver nutrients into that core. You could also potentially use these enhanced vascular organoids for transplantation purposes, potentially for drug discovery, that sort of thing. So there is a drug discovery element. It is a Joe Wu story, of course. But one thing that they're hoping to utilize here to actually achieve that next generation of vascularized organoids and vascular cardiac organoids is an engineering approach, and I'll get into that a little bit. So they used something that I know and love here, which is fluorescent reporters, and I think, of course, you also love these fluorescent reporters as well, Daylon. You have somewhat of a contribution to the paper. I think perhaps you could mention that down the road if you want to. They had 4 different pluripotent stem cell fluorescent reporter systems that they basically commingled in their cardiac differentiation here. And also, very importantly, like I've mentioned briefly, an engineering approach, spatially micropatterned human pluripotent stem cells. Oscar Abules is one of Joe's original cardiac bioengineers who was there when I was in the lab. So, that I'm sure this was his contribution, the micropatterning of these pluripotent stem cells using like a stencil approach to create these really reproducible and scalable vascularized cardiac organoids. And I can talk about it, but I really just encourage you to look at the paper, because you can see the beautiful organization of these vascularized cardiac organoids with the CD31 positive sprout formation at the exterior of the NKX 2.5 and cardiac troponin positive cardiac organoid segment. And then they basically were able to identify this combinatorial micropatterning system, and also a growth factor and small molecule cocktail that when they added that cocktail to the micropattern pluripotent stem cells, was able to, like I said, reproducibly generate this spatially organized, branched, and very importantly, luminized vascular network within a multilineage cardiac vascular organoid that was comprising a lot of the things that you would find in the heart, which is, of course, endocardium, myocardium, epicardium, and even some neuronal cell types. I actually don't know how those got in there, but of course, it is a diff. So you do have other cell types in there as well. I don't know if that has something to do with the nodal conduction system of the heart. Maybe you're producing that in the system as well. But the luminized vasculature, I think, is very important because these are not just CD31, CD144 positive endothelial cells. They're creating tube-like structures, which is exactly what you want for true vascularization, right? You want functionality and some capability there. And then they did the analyses to just show what was being made in these differentiation. And again, very high throughput organoid differentiation here, reproducible with the engineering approach, stencil approach. That's what I really love about that. So the single cell transcriptomics, high resolution 3D microscopy, a lot of different functional analyses to show that these cardiac vascular organoids are structurally and functionally similar to the embryonic equivalent, 6.5 post-conception week human embryonic heart at Carnegie stage 1920. So that's the benchmark that you have to do if you're doing some sort of human cardiogenesis work, is, compare it to the structure of the real thing, if you have the ability to do that comparison. But they also were honest in that they showed that the comparison revealed differences between this artificial system, this vascularized cardiac organoid, and the real deal. They found that Notch and BMP signaling were required for the vascularization in the cardiac vascular organoids, with the inhibition of BMP actually having a more negative effect on vascular formation than Notch. And then they demonstrated that actually the broader utility of that vascularization strategy to use that same vascular inducing cocktail to produce hepatic vascular organoids, HVOs, which are also similarly organized. They have the same branching morphology of the vasculature, luminized vascular network, and integrated with multilineage hepatic cell types. So it's a really cool bifurcation that's happening here, cardiac and hepatic. So ultimately, again, just an evolution of an existing cardiac organoid model system, but I think the vascularization here is pretty advanced. You got the lumen formation, which I think is very exciting. It's reproducible, scalable, answering some of those critical problems associated with organoid production, right? Scalability, reproducibility, heterogeneity. They're hoping to address that using this engineered approach. And honestly, this is something that I want to adapt into my own lab immediately. And selfishly, that's part of the reason I covered this story. I actually reached out to Joe, first, obviously congratulating him on the paper and congratulating Oscar as well. But I also mentioned to him, Joe, you got to put together a protocol because I think that's how this would really help the field. Is there's a lot of moving parts here. There's the engineering stencil approach. There's the vascularization cocktail. And I think having a protocol to kind of lay it all out would be super helpful. So congrats again, Joe. Congrats, Oscar and Adam, on this awesome work. I know it was many, many years in the running for this work. A lot of work that went into to make this a reality. So very cool to see this out finally.
Yeah, this is a beautiful piece of work. You know, culmination of many years, clearly, and a lot of tools, a lot of collaborations. Weirdly, it wasn't just this vascular reporter that I generated when I was in Shaheen's lab that was borrowed or donated to this paper, but also this triple reporter, germ layer reporter, was donated, but not made by me, but made by my postdoctoral mentor, um, Ali Bribonlu. So this really feels all in the family. And the other aspect of that, I mean, I got to emphasize these reporters. It's amazing. I mean, you said there's four reporters, but it's the aggregate of like 8 at least different, reporter constructs. You know, couple of those with these triple reporters. So there's a lot of fluorescence going on there. It's like you're at the disco. But it allows you to see multiple things at once in real time. And that is it's a really beautiful, literally beautiful story to look at, and a really useful tool. I think all of us using these reporters would echo the utility and validate the utility of firm. The utility is in that you don't have to label your cells, right? You have these intrinsic markers that makes the work so much more efficient. And there's a lot of markers that, of course, you can't really label the cells non-destructively. So reporters, can't say enough about them. But what really I love about this story, and this is another connection to my doctoral mentor, Dr. Bribonu, was the micropatterning. And I've worked a bit. I'm working a bit with Idzi Heemskirk, who's by way of Arya Warmflash, a descendant of the Brivonlu lab, and they use a very similar approach here with the micropatterning, subtly different in terms of the stencils and whatever that they use. But it's amazingly powerful, and it allows you to in the case that I was using it, to get really reliable, robust differentiation to primordial germ cell-like cells, which is a very early event, and so is cardiac differentiation, one of the earlier events. But what I was so impressed at here is extending this micropatterning approach beyond these, early inductive differentiation events and getting more into these gastrula stages. So it's kind of taking this scalable micropatterning approach that I think has been used before now. Although I should say, Idzi Heemskirk has really just had a paper in Nature Methods where they extended that to look at gastrulation stage. I don't know if that's out yet. I hope I didn't blow you up there, Idzi. But the point being is that now we're combining these two ideas of the scalable micropatterning and the gastruloid differentiation to really get the throughput and the types of questions you can ask using these models is so much more. There's a much greater breadth. So I love this story. I appreciate how much work it took. I like to see that some of the ideas that we, set the stage for in terms of tool making have really been used to such a beautiful endpoint here. And I just got to say, hats off. Everybody, have a look at this story. 7 figures in each one. You're going to be squinting your eyes looking deep in there because it really is a lot to see.
Yeah, they've really packed a lot into this. And as somebody who you alluded to, the power of fluorescent reporter lines, I I use all these all the time in my laboratory. Seeing is believing, and why not, right? Like if you have the capability just to non-invasive longitudinal live imaging, you should take advantage of it. I mean, a number of these fluorescent reporter lines are commercially available. I mean, I think even Joe's lab also is very willing to share a number of these, but there's commercial sources like Allen Institute where you can get these fluorescent reporters from. Um, so I think it just makes your workflows that much easier, non-invasive. So absolutely incorporate those into your lab. And the stenciling micropatterning is, I think, a very hot topic. Like what you're alluding to, a lot of groups are utilizing this in their organoid differentiations because, yeah, it gets around some of those problems of heterogeneity, and perhaps improves the scalability and reproducibility of these organoids. So a lot packed into this paper, but a lot of technologies that we know and love.
Moving on, we got another story that's emblematic of the amazing progress in terms of structure of stem cell derivatives. And this is from Mary Hutch, who's a friend of the show. who's at the Max Planck, and she's talked about this with us about her liver organoids, but here she extended it beyond. So let me take a step back here just for review. The liver, there's two major epithelial cell types, the hepatocytes and cholangiocytes, but also there's all kinds of other stuff in there, stroma, endothelial cells, and immune resident subpopulations, all of which are really relevant to function and, disease in the liver. And the hepatocytes, when we talk about the hepatocytes, they're unique in that they're polarized, right? They have the apical pole is connected and contributes to these bile canaliculi, which form this continuous network of tubes that are sizable, 1.5 to 2.5 micron in diameter, which, I mean, I know it's not huge, but like that's significant, and enables the drainage of bile into the bile ducts, right? And it's the dysregulation of the flow of bile in these canaliculi, or a dysregulation of the connection between the caniculi and the ducts, canaliculi and the ducts. I'm going to have to say that 1,000 times in this story, aren't I? That the dysfunction in the communication there is what underlies cholestatic liver diseases, which ultimately progresses to fibrosis in many cases, cirrhosis, and liver failure, right? And in the progression of disease, it's the interactions, as I said, between the different cell populations in the liver that are disrupted. And so the idea, which makes sense, is that if you could restore that communication, you could help to, mitigate the progression of disease. And looking at spatial transcriptomic work, there's been a lot of elucidation of like what these cells are saying to each other in different contexts: healthy liver, cholestatic liver. But the interactions are really dynamic, right? So it's hard. You can't like knock in. You can't create an animal that allows you to see and manipulate all these different things. You can, but it's not necessarily practical to look at all of them, right? So it's really challenging to study in vivo and live animal models. Of course, this is the rationale for so much stem cell work. But in this case, it's really relevant, and Mary really takes her tools here to task in addressing that problem. So there's currently no in vitro models that can recapitulate those interactions between those cells. There are efforts, right? You have organoids that are comprised of like either cholangiocytes or hepatocytes, just the epithelial component, because that's kind of a thing about organoids, right? They form these homophilic interactions between epithelial cells, which give them the structure. And there have been the combo cultures of cholangiocyte and hepatocyte progenitors in 2D, but you can't keep those going for more than a week. And of course, the 2D nature of them fails to recapitulate the 3D network and the connectivity of the bile canaliculi networks. And also, they don't have the stroma cells, so you can't really recapitulate the interaction with the stroma of those epithelial cells. So then there's pluripotent derived organoids, right? As you kind of described with those gastruloids of the heart and the liver, like you get the emergence of multiple cell types within those, right? So there's a potential solution, but the problem there is that they remain pretty embryonic, as you well know. All organoids, but the liver, hepatic organs in particular, remain pretty embryonic, and also don't have the same architecture as the adult liver, right? So recently, Hutch Lab showed that there were these cholangiocyte mesenchyme organoid models, but those don't have the other cell types that are in the periportal region. So the Hutch Lab has kind of been marching along here incrementally, and incrementally doesn't capture the scope of the advance here, but moving forward with these large steps here. And in this case, they have this assembloid, right, which is where everyone's going. It's this multi-component organoid system from adult, I should say, This isn't pluripotent or embryonic, and it's comprised of hepatocytes, cholangiocytes, and the mesenchymal cells. And the amazing thing about this is that it recapitulates the architecture of this periportal region, and it models aspects of both cholestatic injury and fibrosis, right? And so the organoids that they were generating were combined with how they make them is they take the hepatic organoids and they combine them with cholangiocytes and fibroblasts. This again, all primary cells. And that mimics the kind of cellular interactions that I was alluding to before, and even show that you can get drainage of bile from the canaliculi into the duct. And the really striking kind of like insight in terms of biology and medicine, understanding of the disease progression, is that with no immune cells, so they don't have immune cells in here. I guess that's what's next for the Hutch lab. They'll add some immune cells, but even without immune cells, just modulating the number of these portal mesenchymal cells that they add is enough to induce a fibrotic like state. So it's just like the load of cells essentially, which I think is a really important insight because it shows that like the imbalance of proliferation of any one cell. It's not just the maladaptive component of cells going to the wrong fate, but growing too much can, I guess that's maybe an obvious idea, but here shown with the in vitro model that shows function and then pathogenic or pathological dysfunction as well. So for me, a really beautiful extension of the Hutch Labs tool set to now have a model that more accurately recapitulates the architecture of these components of the liver, and also the pathogenic disease progression here. So a really cool story. Congratulations, Mary.
Yeah, absolutely. That episode with Mary was one of my favorites. If you haven't already listened to that one, definitely check that out. Yeah, Hutch Lab is doing incredible work on all things related to liver organoids, and yeah, we are biased towards IPS, of course, but we love covering some good primary organoid studies as well. I think one thing you alluded to there at the end was the thing that excited you the most, which is the functionality, and I totally agree with that. I think the bile drainage is really cool. It makes me also wonder, of course, anytime you build the complexity of these things, and build these assembloids going from organoid to assembloid, you gotta there's an element of reproducibility that you have to address. And part of me is thinking, hey, maybe they can incorporate some of the stenciling approaches that the previous paper used to actually get around that issue of reproducibility. I mean, we've had this conversation with Sergi Pasca, one of the godfathers of the assembloid system, and it is heterogeneous. The organoid system itself is heterogeneous, but you combine multiple organoid and tissue types together, it gets even more variable. So it makes me wonder about ways to make this a more scalable, reproducible system. And hey, maybe engineering is the trick.
Yeah, I love that point because uh, you know it reflects two phases of, well, there's so many phases, right? But I think it reflects scientific advance where we spend so much time just like proof of concept, feasibility, and who cares how we get there? FBS, Matrigel, doesn't matter. But nowadays, it really is. I think there's such a premium place on the reproducibility, the scalability, and it's obviously because like we're thinking now as we do the science and make the advance of how these advances can be translated, and why not, while you're developing the system, why not develop it in a way that fits into that more translational paradigm. And of course a lot of that is xeno free and yeah, reproducibility. And again, I have to say it, the micro patterning is so cool because you get the numbers up. I mean, in this, with the case where I was using it, you were going from like 2% of your target cell up to 65, 70%, right? So depending on how you manipulate the system, and of course, maybe there's some limitations, but I agree with you that incorporating all of these advances, into a final product is going to be a really exciting, I think, culmination of all these years of work and all the effort from all these brilliant investigators.
Yeah, absolutely. I think honestly that's one reason why I really respect what Joe does is because of course they do amazing basic science, but they always have an eye to the translational side of things. I mean, he's spun out a number of companies from his lab in very typical Stanford fashion, I guess. But he's got Greenstone Biosciences. Actually, I didn't. I was just looking this up. Oscar, first author of that Joe Wu paper, has his own company now that he's spun out based on his vascularized cardiac organoid work. It's called Bullseye Bio. So there you go. I understand why it's called Bullseye Bio because these little organoids almost look like a bullseye, which is kind of cool. I like that, Oscar. Yeah, but that's we always have to keep an eye on the translational side of things. That's it. The translational world and the translational time is the present. So always important to keep that in mind. Moving on to, since this is a love letter to Joe Wu, apparently this roundup, then the next roundup paper I'm going to talk about is also coming from a Joe Wu acolyte and actually somebody who I briefly overlapped in Joe's lab. This is much more of a old school quote disease modeling approach using iPS derived cardiomyocytes and cardiac organoids, published in Circulation. This is coming from the lab of Shijun Hu, who was a postdoc for a couple of years when I was a grad student in Joe's lab. Shijun is now at the Suzhou University, Suzhou Medical College in China. Maybe we'll actually run into him at ISCCR. Who knows? First author here is Li Chen Ye. The title is Disruption of Cardiac Troponin-Mediated Sarcomere Mitochondrial Communication Results in Dilated Cardiomyopathy. And, one of the first high-profile papers that came out of Joe Wu's lab in the area of iPS cardiomyocyte disease modeling was on the topic of dilated cardiomyopathy, which is a thinning of the left ventricle of the heart, ultimately can lead to heart failure. And back then, and I forget if Shijun was actually the first author of that paper. He might have been about 15 years ago. Back then, they basically created iPSCs from somebody who had dilated cardiomyopathy, a genetic version of dilated cardiomyopathy, and were able to recapitulate the disease to some extent in the individual cardiomyocytes derived from those iPSCs. So it was, very old school, simple. It's funny to think of like 2010 as old school, right? But it was basically just looking at the intracellular sarcomere organization of the cardiomyocytes and saw that, oh, wow, these things are messed up. But now you can really start diving into the mechanisms even more now that we have model systems like these advanced cardiac organoids. And now that we better know how to handle and make these iPSCs, iPSC cardiomyocytes in general. And standardization of iPS reprogramming, right? Anybody, anywhere can basically make iPS cells. And that's part of the foundation of this paper. And one of the dream applications for iPSCs for disease modeling is to recapitulate patient-specific disease and familial disease in a dish. So dilated cardiomyopathy, like I mentioned, has a bunch of genetic factors. My postdoc mentors and the Seidmans were some of the first to identify some of these genetic factors of dilated and hypertrophic cardiomyopathy decades ago. Sarcomere function is really important for causing dilated cardiomyopathy, and sarcomeres are, of course, the individual contractile fibers found in individual muscle cells and cardiomyocytes. Um, and here they were looking at the role of cardiac troponin T, a really important part of the cardiac sarcomere, and how it can, when it's messed up in the situation of dilated cardiomyopathy, it can dysregulate the sarcomere mitochondrial communication in this disease, which ultimately, sadly, can lead to heart failure. So it's an iPS approach. They made iPSCs from a family cohort, I believe in China, who had dilated cardiomyopathy. And there was a specific mutation in this TNT2 gene, which codes for cardiac troponin T. It's an A553G sequence variation. And as you might expect in a disease modeling approach, if you identify the mutation and you see the phenotype in the dish, you got to correct it too. And they did that with the CRISPR. But in traditional circulation fashion, and you're probably familiar with this, Dalen, circulation loves their mouse models. It seems like no matter what journal of the American Heart Association you submit to, you have to have a mouse model, whether it's going from JAHA up to circulation, you got to have the in vivo system, which is we can talk about the frustrations about that offline, but yeah, so they had the mouse model as well. and created basically a mouse that's somewhat equivalent to the human variation. They're able to recapitulate, again, those pathological phenotypes of sarcomere disorganization in the iPS cardiomyocytes. Importantly, cardiac organoids, right? They extended the work to the three-dimensional model and the mice as well. Did a bunch of RNA sequencing, metabolite profiling, co-IP, mass spec to figure out the molecular mechanisms. So they identified this novel pathogenic variant in cardiac troponin T, K185E, as actually the causal sequence variation in this dilated cardiomyopathy family cohort. Um, they saw the messed up sarcomeres. They specifically dove even deeper into the mechanism and saw there was a specific interaction that was messed up between cardiac troponin T and these other sarcomeric proteins called 1433, which causes the dissociation of those 1433 proteins from the sarcomeric structure. And when those proteins are freed up, they do damage and mess things up in the rest of the cell, which they show aberrantly engaged in RAS-RAF1 signaling, messing up P44, P42 kinase activation, which messes up downstream phosphorylation of DRP1. So a whole cascade here, which ultimately messes up mitochondrial fission and fusion. That was the downstream impact of this one specific mutation that happens in cardiac troponin T. Um, and they saw those messed up phenotypes, not just in the 2D cells, but also in the cardiac organoids. The mice had the same problem, and recapitulated some of those cardiac dysfunctions that are found in vivo. So ultimately, it's a disease model, and with a disease model, the other thing that you got to throw in there is a something that can reverse the phenotype. So that's what they did at the very end of the paper, using a mitochondrial fission inhibitor to actually alleviate some of those DCM phenotypes in vivo in the mouse model. Um, Cool study, really cool study. You're taking a single point mutation from a family of people, reproducing it in the iPS model, bringing it to the organoids, to the mouse model, and ultimately figuring out the mechanism and alleviating the phenotypes by applying something in a mitochondrial fission inhibitor that was identified by teasing apart that mechanism. So, real kind of nuts to bolts kind of paper here, again, from a Joe Wu lab acolyte. Won't You can't really expect anything less, right?
Yeah, and how could you ask for anything more? This is really complete as a story. Soup to nuts, I think, is a great analogy. But the as you said it in the jump there is old school being 10 years ago. Just think about all that's incorporated into this one study, which I mean, I guess is a very high bar, which circulation does have a very high bar for publication, but like there's so much work. Um, and I think as you described there, it's incorporating all these modules that were huge steps forward when they were first published and incorporating them into one study that has real meaning and gets to, I think, a clinical translational endpoint that again, it's the story. That's where we are now, right? All these studies now, and with a therapeutic candidate, it seems like. So I am similarly enthusiastic and love this as a kind of representative of the arc of this disease modeling in pluripotent cells, particularly in the cardiac field, where I would argue has been maybe the most modeled cardiac dysfunction and defects using pluripotent stem cells. So another great piece of work here.
Yeah, I would say either cardiac or some kind of neurodegenerative disease, right? Like, well, Alzheimer. I feel like the mechanisms are tougher to model if on the neurodegenerative side of things. I mean, if you're looking at a cell intrinsic defect, I think the cardiomyocyte defects are pretty phenotypically easy to look for. These things are functional and their function is messed up or their sarcomere is dysregulated. So the visual is very apparent with the cardiac disease models. So there's that. Remember back in the day, we're talking about back in the day, right? 20 years ago, you could generate an IPS line and get a Nature paper. And these days, you got to do all this work. It's a new generation, right, Dale?
Yes, I mean those were the days, right, where you would generate the disease specific line and be like, oh, and look, here's the phenotype, and people would give you a standing O, appropriately. But now you gotta like do something crazy. And that's what they did. Another crazy development, which, I mean, who even had it in as a glimmer in their eye? CRISPR or base editing, 20 years ago when we were talking about stem cells, thought we were all that. But here we're talking on a story that goes beyond here with, I didn't even know this was a thing, mitochondrial disease and mitochondrial DNA base editors are. So bear with me here. It's not entirely a stem cell story, but we do this from time to time. And I'll tell you why. It's because this is a tool that is going to be widely used, I think, to add a whole other kind of facet to our study of pluripotent cells here in terms of mitochondrial disease. And not for nothing, but Dolly Lee, who's a senior author on this, had a twofer in Nature Biotechnology with a companion paper to the one I'm going to present, both published in the same issue. So I think that's indicative of kind of the scope of the impact here. But talking about mitochondrial DNA, so 95% of mitochondrial disease DNA, pathogenic mutations, 95% of them are point mutations, right? So a single fix, if we could say that for disease generally, and 95% of genetic disease is monogenic, wow, we'd have a lot more cures. And there's methods for modeling mitochondrial disease in cells in mice. They're elaborate, and complex. And the tools are not really efficient for modeling and identifying the pathogenic mechanisms underlying mitochondrial disease, much less targeting those mechanisms with some kind of therapy. And because there's no curative therapy for mites in every cell, right? How do you deliver to all the cells? And then maybe the manifestations of disease are only in some, but how do you cure mitochondrial disease? The only way now, as you talk about this mitochondrial replacement therapy, which is like an intergenerational therapy, we're talking about at the zygote stage, which has been done on a very, very limited scale and also not really practical for the population generally and the unmet need with mitochondrial disease. So there are these mitochondrial base editing technologies that I wasn't aware of, these cytosine base editors that have been developed based on using good old, remember TALENs? You're taking the transcription activator like effector arrays, so that the TALE part of TALENs, and then coupling that with these mitochondrial targeting sequences, cytosine deaminase, and uracil glycosylase inhibitor. And with that, you can enable the CG to TA conversion in mitochondrial DNA. And that's what they're called, these mitochondrial DNA adenine base editors, right? ABEs. But compared to like CRISPR, nuclear adenine base editors, is very poor efficiency, particularly with the A to G conversion. And this is really important for mitochondrial disease because of the phenotypes that present, the manifestations of mitochondrial disease are dependent on really high levels of mutation above a certain threshold, usually more than 50%, right? So an inefficient adenine base editor, either to model disease or to kind of, mitigate the disease to cure it, you can't have poor efficiency. You're not going to get anywhere. Right. So this was a story that there's a lot of chemistry here, details that you guys should have a look at. But I just wanted to throw it out there because a great new tool. Not going to go into too much detail about this or the companion study, but they made these variants. They screened and created these efficient mitochondrial adenine base editor variants that they reported on how they did it and all that efficiency. And in this study, what they did is they created this Lee syndrome rat model by using and exploiting the high efficiency of the tool here. They get up to 74% of the targeted rats in the F0 generation. So they would inject it at zygotic stage and then let them grow up. They found that like 3/4 of these mice had the disease and it was manifest. And then of course, they made it and then they fixed it. And they took the next generation, so embryos, zygotes from those affected mice, and they corrected the mutation using this precise adenine base editor for the C to T edit. Um, and it restored, I mean, not perfectly, but while they had this up to 91%, they took this one background, or was 91% mitochondrial DNA mutation, and they brought that down to an average of 50. So, you know, pretty significant decrease. Not most, but not all of these patients struggling with mitochondrial disease have that high a level. So bring it down to 50%, I think, speaks to the potential of this approach to, reach a threshold where disease is no longer manifest. I think delivery systemically, or to the places where the mitochondrial disease is likely to manifest, that's still an open question. But like, first you need the tool, right? And I think delivery is a problem across the board. But here, talking about even, in the context of IVF, curing or addressing mitochondrial disease, this is an amazing step forward for a lot. People don't talk about mitochondrial disease maybe because you can select embryos, but like, it's still a thing. It's still a major problem with zero ways of therapeutically addressing it. So this is, I think, a watershed study.
Yeah, very exciting stuff here. I mean, you mentioned that this is a tangential stem cell story, but we're talking about the zygote, of course, the
There you go.
totipotent stem cell.
There you go.
So it's the stem cell, Dalen. Yeah, another reflection on the power of base editors here. And who said TALEN is dead? Right? This is a TALEN-based editing approach. It's really cool to see. I mean, everybody thinks CRISPR is the way to go, but here you go with the TALEN-based base editors. A lot of interesting potential here and applications for base editing these days. Of course, we covered recently that study in New England Journal of Medicine, which is basically using, again, base editors to correct disease-causing genetic variants in the situation of this lipid associated disease of severe carbamoyl phosphate synthase 1 deficiency. Remember, it's like Kieran Musserner and the team, the clinical trial for that little kid KJ, who that was a really beautiful study. But again, just a reflection on the translational power of these base editors. And it doesn't matter if it's CRISPR or TALEN. I think there are a lot of really exciting applications for these technologies.
Yeah, and it feels like it's all happening at once, right? Once you see it in the journal, it's just one case, and then there'll be another, and then it'll be everything. And to see all these different tools emerging alongside, we're talking about tools that are in humans that were developed, almost a decade ago. So just think, with the pace of advance, the things that we're going to be able to do at an accelerating pace moving forward. So this is really exciting. And I'm glad you mentioned that story, because it really puts a human face to the science here, and is a great motivator for all of us. So a really, really exciting time to be alive, Arun. And we're going to talk about that at length at the ISSCR. I think the enthusiasm of all the investigators there is going to be infectious. So tune in for our on-the-ground shows there and all the programming that we're providing. Also, before we get to our interview portion, I have a quick message from STEMCELL Technologies. Are you busy in the lab? Let STEMCELL Science News handle the literature search for you. With 20 newsletters covering immunology and cell biology research, they deliver the latest studies straight to your inbox. Subscribe by July 18th for a chance to win a custom lab coat personalized with your name. Sign up now at www.stemcellsciencenews.com/giveaway. All right, you guys, with us today on the show, we have the director at the Institute for Stem Cell Science and Regenerative Medicine, also senior professor at the Jawaharlal Nehru Centre for Advanced Scientific Research, Dr. Maneesha Inamdar, whose laboratory studies cell lineage, specification, and differentiation during development. In the lab, they've taken a unique comparative approach using embryonic stem cell models, mouse developmental biology, and transgenics, also Drosophila genetics. All of that to decipher the roles of novel genes expressed early in the blood and cardiovascular lineages. The current focus is on analyzing mammalian development using stem cell models as well as clinical studies. Maneesha, thank you so much for joining us on the show today.
Thank you. Pleasure to be here.
Well, the pleasure is ours. It's great to have you on the show. As I mentioned in the intro, you're one of the very few investigators out there that uses a combination of in vitro vertebrate and invertebrate models in their own lab. Why don't we start with an overview in your own words of how and why you use this approach?
Thank you, Dalen, for that question. So I think it's, partly out of necessity, but mainly out of interest. I trained as a Drosophila biologist, and then after my PhD, which I did in TIFR Mumbai, I wanted to move to a vertebrate model system. I didn't know which one at the time. So I took this course at Woods Hole, the embryology course, which of course spanned the whole gamut of invertebrate and vertebrate models. And then I decided I wanted to, work with mice because being a Drosophila biologist, having a genetic model was, very important to me. But at the time, of course, there was not much mouse work happening in India. And, I got a lot of good advice saying I shouldn't switch to mice if I wanted to come back and after my postdoc abroad and set up a lab in India. But that's what I wanted to try out. So I chose the postdoc laboratory, Professor Vicky Boucher's laboratory in Chapel Hill, UNC, because they also had this stem cell model, mouse ES-cell model that could, make the cardiovascular and blood lineages in the spontaneous differentiation in the dish. And I thought I could carry this back to me. And if, my attempts at using mice failed, then I would have something to work with. So that's how I made that transition. And then when I actually came and, set up my lab, of course, as predicted, it took a while to get the mouse work off the ground. It took a while to get the transgenics and knockouts made and so on. So while I was, waiting to make mice, I was, in a manner of speaking, swatting flies, I guess, back to Drosophila, because it was, easy to do, affordable. Yeah. And then, of course, at some point, I decided it's time to move to the human system. There were no human embryonic stem cell lines. This was, 20 years ago. And, it was difficult to know how to culture them. I had made mouse ES cell lines. I had some idea, but we didn't have access to any. So I decided it's time I derived human ES cell lines. So I sort of, with some collaborators, we organized a workshop here in Bangalore. It was an Indo-UK workshop that had the who's who of human stem cell biology come and teach us. And sort of through that, I learned a bit of how to culture the cells and then made a few attempts to derive these human embryonic stem cell lines. This was just about the time that, the iPS cell paper had not yet come out. But it was a time when we did not have any guidelines or regulation or ethics or policy. But of course, one wanted to, derive cell lines that would be used or eligible for use all over the world. So, I was very fortunate to have made contact with the folks at the International Stem Cell Banking Initiative and the International Stem Cell Forum, and they were really helpful. And I sort of figured my way around all the ethics regulation and so on before we had guidelines in place. Yeah, so that's how I ended up making human embryonic stem cell lines, and these are eligible for use, around the globe mostly. They've been deposited in the UK Stem Cell Bank, and yeah, this is really, it's become our workhorse now. So, uh, I think we're really happy to be able to go back and forth between these model systems because, then ultimately it's the scientific question that matters, and we have the luxury of, asking it in whatever system seems most suitable.
Mm.
Yeah, so that's how we got into looking at vertebrate and invertebrate models.
Yeah, you've been a true pioneer for pluripotent stem cell biology in India and around the world as well. And you mentioned some of the systems that you're working with, invertebrate, vertebrate, right? And certainly there are stem cell fate decisions in both Drosophila and mammalian systems that are conserved. And there's a deep evolutionary conservation of developmental pathways, and this is something that we talk about here on the show. These are really powerful model systems, and there are in vitro model systems that are now allowing us to dissect the mechanisms of development in human even more effectively. And you'll talk about some of those works as well, some of these new gastruloid early embryo model systems. But when it comes to comparative biology, how do you think we can best use these different model systems from different species to actually look at those comparisons between different phyla, see how different mechanisms are conserved. Talk more about that conservation of biology and conservation of mechanism across species.
Sure. So maybe I'll take from the example of our experience in this. So, um, when we started off with, of course, this question that interests everybody and that so many people are looking at, how does, a stem cell decide, what its fate is going to be, who decides, what's involved, and so on. And, we were looking at this gene that we had sort of identified based on its expression pattern when mouse embryonic stem cells differentiate into cardiovascular and blood lineages, and found that after being expressed in the pluripotent stem cells, it gets rapidly downregulated and is restricted to the blood, progenitors and the vascular progenitors. So, in fact, that's why we named this gene as Rij, which means blood in Sanskrit. And then later it was named ovarian carcinoma immunoreactive antigen domain containing 1, or OCID1, and turned out to be a family of 2 proteins in vertebrates. So we also then, went on later to show with human embryonic stem cell differentiation that this protein, which localizes to the mitochondria primarily, is also required for human stem cell differentiation, maintaining stem cells, progenitors in multiple systems, including in the Drosophila blood system. So Drosophila has these blood progenitors. It doesn't have true HSCs, but it has progenitors, and we found there is conservation in the way this protein works across species. So you could rescue the Drosophila knockout phenotype by putting in the mouse protein. So yeah, it was interesting because this protein being a mitochondrial protein, one always had the doubt that looking at stem cells differentiating in the dish, and we're seeing mitochondria and metabolism is affected, but that could be due to so many factors, including just having one kind of media or the other. So we wanted really an in vivo model where we could, see how relevant our findings were. And like I said, this protein is expressed in the Drosophila blood system, and later we showed it's also in the mouse blood system and is required to maintain HSC quiescence. So we realized that when one is looking at the vertebrate hematopoiesis, or bone marrow hematopoiesis, trying to figure out what is happening, and if you're looking at something that controls metabolism, there's a problem of limited sampling because HSCs, as we now know, are so heterogeneous, and there are multiple sites in the embryo during development and even in the adult, multiple sites where hematopoietic stem cells reside. So when one takes a small pool and assays them, one is never really sure whether the effects of what you see on metabolism are really can be applied to the whole system. So that's why we turned again to Drosophila, because here we found that, and we could show that the blood development, the hematopoietic organ in the larva, is really this small organ that, well, we first had to deeply characterize it and show that it too has this heterogeneity in the blood progenitors. But the beauty of the system was you could look at the entire hematopoietic organ in a single animal in, two or three two or three images side by side. And you could look at the developmental timeline going from anterior to posterior. And of course, it's an excellent, powerful genetic model. So you could really test whether, whatever you see actually affects the animal, makes a difference to it or not, and use genetics for it. So that's how we ended up going back and forth. And I think that was the real power of the approach, being able to test in vivo, in the mouse, in the fly. And of course, now with the human stem cell models, one can hope to go, a step further.
Yeah, listening to you, I am appreciating more and more the scope, the breadth of your work. And I think it all starts from the beginning, right? As you described, moving through those different model organisms, you get this comparative view, right? This kind of Evo-Devo
Right.
overview and framework to your thinking. And there you're describing a similar thing of the interrelationship and how you incorporate these multiple models to elucidate, fundamental mechanisms and processes in biology. And of course, you're talking about hematopoiesis. Any listener to the show knows that really piques my interest. And that's a major focus of yours as well, right? Hematopoiesis and cardiovascular development. And you had this story that I read not as deeply as I would like, so I'm going to ask you to fill in a little bit. But it was a similar idea, kind of, in terms of like this e-life story that you just had, in terms of like the interrelationship of all the signaling and the architecture, right? And specifically here, TGF-beta signaling and how it relates to microtubule stabilization, and then how that impacts vascular patterning, this kind of meta thing, which is universally important. Can you fill in a little bit on that and that idea about like the interrelationship of all the cellular components that are appreciated mostly as one thing, transport or structure or signaling, whatever, where in fact these things kind of have multiple roles. Can you talk about that a bit?
Yeah, sure. So a bit of background. So when we started looking and trying to answer this question about how do cells, make fate decisions and all that, and I said earlier, took this expression-based approach. I mean, if something's at the right place at the right time, maybe it's relevant for, directing something. And we came up with this other molecule, which turned out to be a cytoskeletal protein, again expressed in a very restricted number of cells during ESL differentiation in the sort of blood lineage. So we named this Rudhira, which, as you might guess, means blood. And so somehow we ended up looking at organelle proteins and not at the, sort of classical approach of looking at ligand or receptor, signaling molecule, or transcription factor. And it was really challenging to figure out how these proteins might do anything other than quote unquote housekeeping roles. But there again, having genetic models really helped us. And specifically, we found for Rudhira that it is cytoskeletal protein that binds tubulin as well as binds vimentin, so it acts as a crosslinker, and it's very tightly regulated. So this is a gene that's conserved through evolution, but in humans, it is associated with many hematological neoplasms. It maps to chromosome 17, to a hotspot of several cancers, actually. It's a very large gene, over 900 kb, but the transcript is just about 3.6 or something kb, so huge regulation at the intronic level. Nevertheless, the protein is quite unstable, but it seems to have this function of binding and stabilizing microtubules, and its expression is transient, rapidly goes up and down. And what we could show is that this is essential for cell migration. So when you give a migratory cue, for example, doing a scratch wound assay, the protein rapidly relocalizes to the leading edge of the migrating cells. And this is essential for downstream CDC42 activation and reorganization of the cytoskeleton. So when we made the knockout mouse and looked at the phenotype, we saw that, as predicted, there's a cardiovascular defect, there's a patterning defect, and that is because though endothelial cells are specified, angiogenesis is not happening properly. The hierarchical patterning that you see in the vasculature is perturbed because cell migration and is perturbed, and angiogenesis, of course, as you know, requires cell migration for the patterning. So then we were looking at what might regulate this, and looking at the transcriptome, we found that TGF-beta signaling seems to be perturbed, which is, of course, very well established that TGF-beta has a role to play in vascular development, cardiovascular development, and so on. So we started looking at the cell biology of it, and looking at what happens to TGF-beta signaling, and what we found that surprisingly, there seemed to be perturbation downstream of the receptor activation, and we saw that if we have a knockout of Rudhira, then the SMAD activation levels are affected. And there is in the literature a paper from long ago that showed that SMAD activation, phosphorylation of SMAD, depends on it getting dissociated from the microtubules. So it's normally present, bound to the microtubules, and when TGF-beta ligand, activates signaling, you get SMAD dissociated from the microtubules, but the mechanism of how this happens really was not known. So based on what we saw of what Rudhira does and how it binds and stabilizes microtubules, we then tested whether it's responsive to TGF-beta signaling, and whether it's binding to microtubules and SMAD activation changes. And what we could show that indeed, our model is that Rudhira binding we can show increases upon TGF-beta activation, and we think this is what displaces SMAD from the microtubules, because we can block one or the other, block transcription, translation, and so on, and see that these effects happen. So there is a dual role. One is a sort of fast role of binding to microtubules, and then there is a transcription dependent role where it actually also activates TGF-beta signaling. So there's this sort of complex network, and we are really sort of excited about this, because this is a role for a cytoskeletal protein, but it's a very signaling level role. And something like the microtubules or the tubulin cytoskeleton, which is present everywhere, we think molecules like Rudhira can give a context dependent regulation. So one can now think of ways to regulate by targeting Rudhira.
Yeah, really beautiful mechanistic story there. And we talked about the power of in vitro and in vivo model systems to elucidate some of these mechanisms. You're also, like we mentioned earlier, you were a pioneer in generating some of the first embryonic stem cell lines and are a pioneer in utilizing in vitro model systems for these basic mechanistic discoveries. Your website also mentions your recent work on gastruloids, something I mentioned at the top of the interview. These are, of course, incredible models for studying early human development and opening up that black box of human development in a way that really hasn't been possible, both human and just mammalian development in general. So, how do you see the role of these gastruloids and organoids and other 3D model systems in further progressing developmental biology? Could you talk about this next generation of model systems out there?
Yes, I think it's a really exciting time, with all the various organoid models. Organoids, of course, have been used for some time now, and for various sort of tissues and organs like the intestinal and the brain and cardiac and so on. But the last few years, I think the mushrooming of the various organoids from the pluripotent stem cells that model really early events in human development. I think that has really opened up a whole new area and a new dimension. And the reason we got really excited about that, because now, like you said, that lets you look into this black box of early human development. And being a developmental biologist, that really really excites me. So what we are doing is, we're trying to understand how mechanisms such as what I described, looking at the mitochondria or cytoskeleton, how those may regulate, or what is the status of these kind of regulations in early human development, because there's so little we know, but we know enough to realize that even though you have a mouse as a mammalian model, it's things can happen differently. So that is really an open field that we are interested in. And also in terms of a practical application for translational work and so on, because while one is very aware that given the sort of limited number of people that we have in India working in stem cells and limited resources, it's important to do something that also has application in the near term. And it's always been a little sort of scary thinking of having cortical organoids or cardiac or other organoids that take weeks and months uh, to make. That takes a whole different level of resources for any kind of scale up and screening. But the early embryo models, or aspects of embryonic development that are modeled with gastruloids, blastoids, and the like. I think these, since it's just a few days that one needs to model them, that's something we realized would be a really good avenue for us to pursue. And that was really catalyzed by, we started sort of a program funded mainly by the Gates Foundation, for which we got funding last year, called the Program in Women's Reproductive Health, or WORTH. And the idea behind this is to make, have stem cell based applications that are aimed at facilitating solutions that'll help towards women's reproductive health and choices. And the Gates Foundation has this large program with many, institutes and centers across the globe in the area of non-hormonal contraceptives. And our goal is to use these kind of models, to see if we can have an assay that will de-risk any kind of molecule by building teratogenicity assays, and that's where our sort of current focus is. And we've sort of been improving or trying to establish these models that have been pioneered by so many others in the field, to see whether we can make these reproducible and robust. And I think most importantly, we are making sure that we use Indian origin stem cell lines, ES or IPS, that are from the Indian genetic background, to sort of make these models and just study so many aspects of early human development, but also the impact of the genetic background, if any.
Yeah, and that's such a key point. I mean, you've been an innovator in the field, period. But I think in particular, you've been an innovator in India and a proponent and advocate for, really showcasing and highlighting science and biological material of Indian origin. I mean, that the making another stem cell line in the US was more, I developed the first stem cell line in New York City, but who cares? I mean, you developed the first stem cell line that actually provided stem cells that represented Indian genetic diversity. It was an important and critical contribution, and that was approaching 2 decades ago. But you've continued on that path, right? You've been an outspoken advocate and an opinion leader, and you've been a part of a lot of these discussions with the WHO, also ISSCR, regulating stem cell research, but also gene therapy, which we haven't really talked about at all on this show. It's a stem cell show, makes sense. But I mean, you have this whole other aspect to your work about the impact of gene therapy. And you have a unique perspective, right? You represent a whole group of people in a part of the world that I don't think a lot of people think about, right? So tell us about that experience in terms of like being a part of those discussions at the WHO and the ISSCR with those groups of people, and what do you think are the real critical gaps there to fill in terms of like providing a more diverse, not just group of scientists, but just addressing the needs of a diverse group, of all people.
Yeah, sure. Well, that's a lot to pack in, but let me try. So, as I said earlier, when I had to derive the human ESL lines, since there were no guidelines and so on at the time, I was really fortunate, to have the help and support of so many people across the world, so many experts who were very generous in offering advice and expertise, and also very inclusive in terms of inviting me to these meetings. And so it's getting close to 20 years that I've been going to the International Stem Cell Banking Initiative meetings. And though we didn't have a stem cell bank, just to learn how should I bank the cells, because I've been providing the cells over the years to so many people, and make sure we have the quality control done properly. Though we at the time we didn't have the resources or the wherewithal to do it. So that's how I sort of got interested in this, and made sure to educate myself. I used to go for the meetings of the ISCBI and the ISCI, and was involved in one of the ISCI-2 projects of the looking at ethnically diverse lines and coming up with standards for ES and IPS cells. So we were very fortunate that our lines were included in that study, and so that sort of made me sort of acutely aware that there aren't many others in India who are involved in this, and we need to increase the critical mass of people. Of course, in due course, we got our own ethics guidelines, which are very good and modeled on the other guidelines from the UK mainly. So yeah, this sort of got me interested in the ethics, the policy aspects, because I was requested to contribute to some of these discussions, even nationally, and then I had the opportunity to be on the WHO committee for developing the guidance framework uh, around human genome editing, and this was soon after the reports from China about the CRISPR babies. So yeah, it was a bit of occasionally feeling like the imposter, because I'm sort of representing this huge uh, number of people, and it was really critical to me, to convey to the rest of the committee members or community that there are these diverse perspectives, and I may be, representing only some aspects that are sort of based on my experience, but I still thought it was important to voice, whatever those sort of ideas or concerns might be, because it was really critical to get the opinion of, like, if you consider the global south, not just India, the global south is hardly represented in many of these discussions. At least it was not till a few years ago. So it was really important to be part of these discussions, voice my opinion. And I used to say, I sound like a broken record, putting forth the same 2 or 3 points again and again and again, because, though every committee that I've been in was very well intentioned, very acutely aware of the necessity for, having diverse voices, often, unless one has experienced it, you really don't know what the issues are or what the problems are. So, for example, one issue I find is language. Most of the guidelines or regulations that committees make are of course, in English, but maybe translated in a few languages. So, for example, the official 6 languages of the WHO, but at least half the world doesn't speak these languages. So it was very important to involve local governing bodies or offices of, let's say, the WHO or any other agency, the ISSCR being an international society. Uh, you know, it's important that whatever messages about stem cell regulation are conveyed in local languages. So there's a big component of public education and engagement and empowerment also that I think is something that I would like sort of to propose that ISSCR and other organizations also take up. Yeah.
Yeah, I mean, these are extremely important considerations for a field such as ours that is so diverse and is so globally represented. This is we have the International Society for Stem Cell Research, right?
Right.
It's not the American Society, it's not the Indian Society. This is a global society with global science happening in across every corner of the world. And as somebody who has mentored students across disciplines and from different backgrounds and has spent time doing science in different places worldwide. Of course, you were just down the road from us at Duke. Both Dale and I went to Duke for our undergrad.
Right.
We won't judge you for spending time at UNC. It's a wonderful place. I'm sure you're aware of the rivalry there. But what do you believe are some of the essential skills or mindsets that young stem cell researchers need today, especially those from underrepresented regions around the world? What are some of those critical skills that they need?
I think, the critical skills are really the same as for any other field. First of all, a real, passion and interest. And of course, I think there's no substitute for dedication and hard work. I don't think there is any special or magic formula for stem cell biologists. But I always caution students not to get caught up in the optics and the glamour. It's just sheer hard work, I think, and of course a passion for the field. So I think, but I must say we are now at a stage where we've progressed a lot. There is so much opportunity in India in the field. So it's no longer a limitation. And actually, if I may, keeping this in mind, at InSTEM, we've started a program called ESCORT, which is actually funded by the Department of Biotechnology. And ESCORT stands for Enabling Stem Cell and Organoid Research and Training. And we've invested a lot of time and effort into establishing state-of-the-art facilities where we routinely hold workshops. In fact, we have tied up with the International Stem Cell Banking Initiative for our workshops. So we have a lot of, participation in terms of teaching and mentoring from there. And so we've set up this facility where people can come and train, or they can come and work, because having trained many people, I know they train, then they go back to the laboratories, and sometimes they don't have the wherewithal to set it all up. And also someone may want to ask just one question in their research using stem cells, so they don't need to set up a whole lab for it. They can come work with us collaboratively, or we have various models for academia and industry. So so that's what InSTEM has set up now. And I think for young people, the outlook is really very good.
Outlook is great, I think, for all of us, but young people in science in particular, and in India, I think. And I think that's a point of emphasis. We talk about science globally, but science in India is a big deal. Like many things, many industries, and the output of India across the board is burgeoning. At the Jawaharlal Nehru Centre for Advanced Scientific Research, which is in Bangalore, India. That's where you are. It's a relatively young institute, right? Established less than 40 years ago, but recognized right now as number 7 among the world's top 10 research institutes and universities by Nature in a normalized ranking of high quality output. I'm not one to put a lot to like lists and all that, but this is like a list from a highly prestigious scientific journal that was normalized, whatever that means. And the reason I mention it is just because all the other places on that list took about an average of like 400 years to get to that point. What is it about the center that makes it so great to do science there, and what accounts for that amazing pace? Is it investment? Has there just been a latent potential there in India that is just now being tapped? What do you attribute that amazing progress at the JNC to?
Well, I think it's a mixture of everything. First of all, the institute was set up by Professor C.N.R. Rao, pioneer, one of our best scientific minds, and the way we do science, we don't consider ourselves to be somewhere far away in one corner of the globe, but it's really cutting edge science, and supported in so many ways by the institute through national and international funding, but also through government funding. So there is, I think, a lot of political will to take science forward and to really push science. So for example, the Department of Biotechnology, under which InSTEM, where I am currently, comes, has really helped launch this Bio E3 policy of the government of India, which is basically for taking biotechnology forward, and making sure that we keep the science, but also the environmental aspect and the ecology, and the bioprocess and bioproduction. So there is quite a big thrust, and I think our education system also gives us, really the best students. I think we are really, really fortunate because, everything that I'm talking about here is, done by excellent, excellent graduate students. And we're, really fortunate to have these young, bright, enthusiastic minds that decide they want to do a career in research.
Mm.
Yeah, I mean, as somebody of Indian origin, I was actually born in Bombay before moving to the United States. It gives me such pride to see the growth of biotech in India, and India has always been just a powerhouse in science, for many, many years, even before the biotech revolution in physics and so on. My dad actually trained at the Baba Atomic Research Center in
Oh,
Bombay
wow.
many years ago. So India has been at the forefront of science for decades and decades now, but it's on the biotech side of things, the growth is really astounding.
Yes.
So how do you see India's role in evolving the global stem cell and regenerative medicine ecosystem over the next 5 to 10 years? Given the growth of the ecosystem within the country, how do you think India is going to establish its leadership on the global stage?
I think, there is really a big push to sort of take the field forward and, propel it forward. And that is, from the government funding, from industry, from philanthropy, because it's very clear even to the lay person now that, stem cells really are the future of medicine, and future is almost here actually. So in the next I think 5 to 10 years, what we are going to see is really, you know an expansion of all these efforts, because through platforms such as the ones that I mentioned, that we are building for training, technology advancement, and especially using sort of areas where we can tap into a niche or a potential that maybe we can do uniquely, that may not be possible in many other places, such as, like I said, using stem cells of, representing our Indian genetic background. And as you know, there's no one Indian genetic background. There's a whole variety. So, already at InSTEM, we've established very recently the biobank. We already have, from projects done earlier uh, at InSTEM with our collaborators, over 100 iPS cell lines that were derived, from families with a history, deep clinical history of mental illness, for example. So that there are control lines and patient lines. We have ESL lines, like I mentioned. We have, a bank of HLA matched GMP grade cell lines from the Center for Stem Cell Research, which is actually the clinical translation arm of InSTEM. And so we already have all these lines, and we're already developing assays. And one of the things that we have done is, to sort of projecting for the next 5 to 10 years, established what we call CREATE, the Center for Research, Application, and Training in Embryology. So the idea being is, we will develop these models of early development, human development, as I said, use preferably cell lines, including cell lines of Indian origin and other ethnicities, and make robust pipelines that can be used for drug screening, for understanding deep biology of early development. So you can imagine that that really opens up a whole new suite of applications that can be scaled up very rapidly for both academia as well as industry and clinical translation.
Wow, future looks so bright. And I mean, you've played a large part in building out that infrastructure and establishing, the foundation for that work. So congratulations on your illustrious career to date. And listening to this, we're coming to the end here, but I had a couple ancillary questions here to ask, science related. And listening to you talk about your work and your career, it seems like you're someone you followed the thread, right? You've got a curious mind. One thing has led to the other, and you've not been confined by the invertebrate model or by the non-human model. You've gone right to where your interests lie. So I think an appropriate post-game question here for you would be like, what if you weren't a scientist at all? What do you think you'd be doing with your life if you hadn't gotten on this track?
Well, I would probably, I like sort of looking deep into development. So I'd probably be some kind of surgeon or something where I can look inside and see what's happening. But apart from any kind of, academic interest, I probably would love to be somebody who can tell a story or entertain, maybe theater, something, that I would have loved to do and sort of, tell a narrative and connect.
Well, I would love to hear a story from you, Maneesha, but we'll have to wait till after the show. Finally, I would like to hear this. What's the best piece of advice that you've ever been given or that you've ever given yourself, either professional or not?
Yeah. Sort of one encounters so many different situations and people and so on. It's very easy to get excited, but I think some of the best sort of advice, as well as sometimes I experienced, is that it's best, not to get agitated too much about things unless it's science. I mean, you can argue about science, but if you spend too much time arguing about other things, I think it takes away precious time from your science. Is some of the advice that I heard, and also in terms of sort of practically practicing. Managing people and so on, is best not to take. So I heard about something called HALT, HALT. So don't take decisions when you're hungry, angry, lonely, or tired. So, one tries to remind oneself. Yeah.
Well, then I should never be making any decisions, quite frankly, but I think that maybe my wife would tell you she already knew that. But that is a very good point. Don't get too agitated. And it's all opinion unless it's science, right? In our respective countries, India and the US, the political climate has gotten very supercharged. And I would agree with you. Why focus on opinions and beliefs when you can focus on the objective truth that science offers? So if we can all keep our heads on that ball, at least the scientists amongst us, then we can increase our productivity and get a meal in you, get a good night's rest, and find a partner, and whatever the A was before you make any important decisions. Thank you so much, Maneesha. This has been really delightful. And I hope to see you at the ISSCR or some other meeting in the future.
Sure. Thank you, Dalen. Thank you, Arun.
All right, you guys, that brings us to the end of today's episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and roundup papers. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELL Podcast, or via email at info@stemcellpodcast.com with feedback or to suggest guests.